Fourth in our genetic age panel discussion series. I'm Robert Mittman, and I am the facilitator, moderator for the session. I'd like to acknowledge and to thank our sponsors and conveners for this session, from the AAAS, Barbara Jasny, and from Affymetrix, Katie Buck. Both of them labored mightily to get these panels all put together and to give us a through thread of content that has so far today been quite interesting. If I can recap the three panels that went before, we had this morning an initial panel that was focused on the organization and funding of genetic research. And if I had to pull a single conclusion from that, it would be that increasingly we're going to see biological research, genetic research done in cross-disciplinary, cross-institution, cross-sectoral teams. A larger share of it will be done like that, which will lead to a need for greater amounts of collaboration. Our second panel was focused on the information technology and intellectual property infrastructures that will support genetic research, and it was actually quite a nice response or foil to the first panel, in that we were able to investigate quite a bit to what extent technology will be able to overcome some of the fragmentation and compartmentalization that can result from this sort of cross-institutional research. And as well, we looked at some of the issues in patents and especially in open source approaches for managing intellectual property or sharing intellectual property. The panel that just preceded this one was on the economics of health, and it was quite a wide-ranging panel, ranging from long-term demographics to the health system in Sweden to a 750 doctor group practice in Wisconsin that is highly automated, makes a lot of use of electronic medical records, and is building an 85,000 person research database that includes increasing amounts of genetic information. That last bit about having databases about populations that include genetic information raised a number of fairly interesting policy and ethical questions that have to do with the use and application of that genetic information. And luckily enough, we have a panel here that's prepared to talk about a number of those issues. The focus on this panel is going to be on regulation and policy and public confidence about genetics, genetic research, and the fruits of genetic research. Before I introduce the panel and begin to dive into the questions, let me say a couple of words about format. This session is going to be light on presentations and heavy on discussion. What I will hope to do is to stimulate discussion first among the panelists and then eventually with all of you as well about the issues that are in question. So I'll be circulating in the audience with the mic, and I hope that you will be ready with some questions and comments to the panelists. Let me introduce the panel. Starting on your left is Tom Murray. Tom directs the Hastings Center, which is a center that's focused on medical ethics and the ethics of genetics. That's it. I just kicked my own power switch. I think that's sonic seppuku there is what that was. Next to Tom is Vince Bilker. Vince is at the National Institute for Standards and Technology, and at NIST works on some of the standards that have to do with the infrastructure for genetic research. He'll say more about that. Next to Vince is Kathy Zune. Kathy is formerly with the FDA, so has had a hand in regulation there, and is now at the National Institutes of Allergy and Infectious Diseases, also working on genetic research, and is very active in some of the standards efforts for the underlying technologies for genetic research. Next to Kathy Zune is Kathy Hudson, and Kathy is with the Genetics and Public Policy Center, and she'll say a bit more about that center and the work there. And then finally, on my far left, is Barbara Koenig. Barbara is at Stanford University and looks at these issues of genetics and policy at Stanford. What I'd like to do before I launch into the content for the panel is first to ask the panelists each to introduce herself or himself in a little bit more depth than I just did, so that you've got a little bit more of a sense of their point of view, and as we talk through the various issues that you know where they're coming from a little bit more. And Tom, maybe you can begin for us.
Okay, this is without warning, but it's fine. Tom Murray. I work at the Hastings Center, which is a nonprofit, nonpartisan research institute, now in its 36th year, if you can believe it. And it has been feisty and struggling for all 36 of those years. So we're always glad to show up the next year. Glad to be here. My own interest in genetics goes back a long time. We did a project at the center about 25 years ago on genetic screening in the workplace, trying to anticipate the uses that might be made and to make some policy recommendations, which I should know were all violated by the Burlington Northern Railroad Company when it adopted its program of surreptitious genetic testing in the workplace. If they'd read our papers, they would have saved themselves a lot of trouble. We now have a project at the center on new issues in newborn screening, and I think that's what I'll be talking about later.
Thanks. Vince?
I'm Vince Bilker. I'm Chief of the Biotechnology Division at NIST, the National Institutes of Standards and Technology. We're not a part of NIH. We're associated with the Department of Commerce. We're an infrastructure agency. We're the measurement people. I've been made aware over the last couple of years since I joined NIST that NIST is over 100 years old as opposed to 36. And in many ways, NIST was established to capture the commercial value in the fundamental discoveries that were coming out of physics at the turn of the last century. And I think Congress, the political and the scientific programmatic people were wise in those times to recognize that certain fundamental quantities needed to be at least consensus standards, or there needed to be agreement about what an ohm was or what a volt was going to be. And that gave birth to what NIST has been historically. If we look upon not the genetic age, but the age of biology for the next 100 years, there are going to be measurement problems that will be just as important in moving the discovery aspect of biology into the marketplace and into the delivery of healthcare. I think throughout the day we've been hearing about paradigm shifts. The one that I recognize, the one that I think my agency needs to respond to, is the paradigm shift which says biology is becoming a quantitative science. The discoveries and the implementation of those discoveries is going to depend as much on the ability to quantitate information as it will be to classify it and to qualitate it. So that's why I'm here.
Good, thanks. Kathy?
Yes, I'm Kathy Zune, and my current position is Deputy Director for Planning and Development of the intramural program of the National Institutes of Allergy and Infectious Disease. I've had the good fortune of being there now approximately 8 months, and prior to that I was for about 1.5 years with the NCI. And it actually is a nice dovetail into my experience of 23 years at the FDA, where I spent 3 as the director of the Division of Cytokine Biology and 10 as the Center Director for Biologics. Over those years and continuing now, I've had an opportunity to work with a number of standard setting bodies for various technologies, including looking at genetic testing and in a variety of senses and proteomics, so gene expression microarrays and proteomics. And it's been important, I think, and is important for the furthering of the field to really get a handle on standardization of this technology, and so that's one area. Also, in terms of regulation, this is a new and emerging area, one which FDA has not had a large experience with, so it's going to be an iterative function, and I believe has been an iterative function over the past, I would say, 7 to 8 years for both the proteomics and looking at many of the genetic testing, and that would include all of the major centers within the FDA. And so I'll represent a small portion of that because I'm no longer with the FDA, but having been involved in those issues, I think I can at least frame those. And finally, I have my own research program in which I use both gene expression microarray and proteomics, so I'm interested in, on a personal level, in all of these issues. So thank you.
Good. Thank you. Kathy?
My name is Kathy Hudson. I came to Washington many, many years ago as a AAAS Congressional Science Fellow, having gotten my PhD in molecular biology. I then spent about 7 years as the Assistant Director of the National Human Genome Research Institute through the near completion of the Human Genome Project, and then founded a center at Johns Hopkins University to look at what kinds of policy challenges are being raised by advances in genetic testing and genetic technologies, and to try to provide the tools that policymakers will need as they think through these challenges in the weeks and years ahead.
Barbara?
I'm Barbara Koenig. I'm at Stanford University, as was mentioned. And just to make clear my disciplinary perspective, I'm a medical anthropologist who works in biomedical ethics, but I work really from the perspective of an anthropologist, which will become clear, I think, as I comment on some of the issues we're discussing. In terms of my past experience in research, I think perhaps the reason I was asked to be on this panel is because during the Clinton administration, I spent about 3 years serving on a federal advisory committee called the Secretary's Advisory Committee on Genetic Testing. And one of the goal of that group was to make recommendations about oversight of the introduction of new genetic tests in the US. That was a very interesting and informative exercise. We did not accomplish everything that we wanted to in terms of instituting some level of FDA review, for example, of the introduction of new genetic tests. In addition to that sort of policy work in Washington, I have done a fair amount of work on issues in the new genomic age myself as an anthropologist. And Kathy Hudson and Tom Murray and I were saying that in some ways we reflect the range of the field, in that Tom is working on newborn screening, Kathy's working on reproductive issues, and I've worked more on issues of adult health and medicine, and how genomics is going to affect adult medicine. I've worked on genetic testing for breast cancer susceptibility, as well as genetic testing for Alzheimer's disease risk, and did a project 5 years ago at Stanford which we sort of locally talked about. We used the sort of title "Individualizing Medicine Through Genomics." So those are the kinds of concerns I've had throughout my academic career. And I think the other thing that perhaps distinguishes my approach is that in my own academic work, I tend to be interested in the issues which are very difficult to regulate, or the sort of social consequences that are really important to consider, but are less amenable to a standard regulatory approach.
let me frame the session a little bit before I turn to the panelists. It's necessary if we're going to really benefit from the possibilities of genetic and genomic research, that the public be appropriately involved, that the public be sufficiently cautious so that scientific excess doesn't take us into dangerous territories, and conversely, that the public be sufficiently open to innovation, that we don't prevent potentially beneficial treatments from being developed and applied. So ultimately, a lot of this is about public confidence. What are the mechanisms by which we affect public confidence? Well, clearly, the press is one mechanism. We don't have the press represented here, although we actually did on an earlier panel. The press, though, is really just a communications channel that reflects some work that's going on in a number of other areas. And some of it has to do with the extent to which we're thinking appropriately about the ethics of the science that we're doing, the technologies that we're developing and applying, and as well that we have an appropriate level of regulation that promotes efficacious technologies and prevents safety mishaps. This panel is going to really take place in three chunks. The first bit that I would like to talk about is just public confidence. What is the level now in the opinion of the panel? And I'm going to give you the heads up, panel, that the question is coming. In your opinion, is the public overly cautious and preventing beneficial things from coming to bear? Conversely, has the public overly bought into the hype, because frankly there has been a fair amount of hype about genetics and expecting too much? So first question, level of public expectations and confidence. Second, I'm going to actually follow what Barbara observed, that we have people looking at three parts of the life cycle of humans and ask them to comment from the point of view of newborn screening and reproductive health and adult health, what appears to be the impact of genetics, genetic testing, the discrepancy that we have at times between the power of our genetic tests and our power actually to treat for the diseases or predisposition to diseases that we can detect. And then the third piece is focused on making sure that the technologies that we're developing and bringing to market are appropriately regulated, that the standards and regulations around those are correct. And so we'll hear from Vince and from Kathy Zun to talk about those. So let me start with that beginning question that just has to do with the level of public confidence and where is it now, where do you think it's going? And Kathy Hudson, I believe you've got some information and some material to report on that.
And do you want me to do that now? Or
if you'd like to do that now, that would be great.
This is in the context of reproductive genetic testing, but can I make a comment first?
Absolutely.
I think when you ask about the level of public confidence in genetics, it really begs the question, what's confidence and what's the wholesome level of confidence that we might hope to see? And so maybe we as a panel can talk about that. I'm going to say just a few words about what we know about the public's views about genetic testing in the reproductive context. Let me see here. So we've talked a lot in the sessions before this session about the various prospects for genetics and the promise of genetics in medicine, and certainly the area where there's been the largest clinical application to date is in genetic testing. And the area of genetic testing that has touched the most people in America is in the reproductive context. So it's We've seen a tenfold increase in the number of genetic tests over the last decade, and this steep trajectory is likely to continue for many years to come, I would expect. So we can now apply those genetic tests virtually any time in the human reproductive cycle. We can test parents to find out if they're at risk of passing a genetic mutation or genetic characteristic to their children. We can do genetic testing on embryos produced through in vitro fertilization, and you can see the individual cell being slurped off of that early human embryo, and that single cell will be tested and be the basis for deciding which embryos to transfer into a woman's uterus to try to start a pregnancy. We can do prenatal testing to identify genetic characteristics of a fetus during pregnancy. And as Tom will talk about, most newborns in this country are tested at birth. So the debate about policy, particularly reproductive genetics policy in the United States, has been very polarized. On the one end are technology enthusiasts who never met a technology they didn't like and never really met a regulation that they did like. And at the other end are mostly conservative religious voices that are deeply, deeply concerned about any technology intervention in the reproductive process, particularly if it will involve harm or destruction of early human life. So as a consequence, we've ended up in a situation in which the policy environment has been polarized and, I would argue, paralyzed. And so we really haven't been able to see the policy keep pace with the science. So what are the sources of potential policy? Well, there's federal sources, there's state sources, and then, of course, professional societies can self-regulate in this arena. And on what basis? What sorts of goals might we have for the policy process? Well, we might want to set ethical standards for the use of these tests. We might want to set safety standards for the use of these tests. We might want to govern access, and we talked in the earlier panel about the use of this information by insurers to deny access to health insurance. And then we might want to have regulations about the quality of care that's delivered to patients. So we have put together a report that's really in the model of the Office of Technology Assessment, which some of you may remember, which presents options, lays out what are the policy alternatives and what's the good, bad, and ugly of each of those policy alternatives in order that as policymakers in all three of these realms, the federal level, the state level, and in the professional sector, are able to think through what are the possible alternatives and what are the likely implications of each of those policy approaches. And copies of this report are on the back table, so please feel free to pick one up on your way out. So at the same time that we were developing these various policy options, we also wanted to understand where the American public is. What are they thinking and feeling about these technologies? I'm just going to provide a couple snippets of that information. So we asked people about, how do you feel about reproductive genetic tests? Do you approve, disapprove of these tests? And we ask about prenatal genetic testing, which will be in green, and testing of embryos, pre-implantation genetic diagnosis in yellow. And we ask about a number of different applications. What about for a fatal childhood disease? How do you feel about that? And you can see that a majority of Americans say that they approve of that application. A similar majority also approve of using reproductive genetic testing in order to identify a fetus or an embryo that will be an HLA match for an older sick sibling and be able to provide tissue to help treat that older sibling. Adult onset risk of disease, such as breast cancer predisposition or other forms of cancer, similarly high levels of approval, and then you see it dropping as you ask about using genetic tests to determine sex. And then hypothetically, and we certainly can't do this today, if we could do genetic tests that indicated non-medically associated traits, how do you feel about that? And just for comparison purposes, we ask about cloning, reproductive cloning in red, and research cloning in white. So if you were to ask sort of where the general American public is and sort of where they draw the line, you would say sort of between these health-related applications where a majority are approving of these uses and non-health-related applications where a majority are disapproving. So we also ask people about their views of regulation. So if there's a line to be drawn, who should be drawing that line? So we asked for prenatal genetic testing, and I'll show you also for pre-implantation genetic diagnosis. What's the federal government's role? Should the federal government ban this stuff, regulate safety and ethics, or keep their hands off? And you can see that there's no slice of this pie that's more than 50%. There's no clear-cut answer or sentiment out there in the American public about a specific role for the federal government, unless you add up the blue slice and the yellow slice. Safety, those who think the federal government should regulate safety, and those who believe the federal government has a role both in safety and in setting ethical standards, and you get a majority of 68%. The distribution is fairly similar for pre-implantation genetic diagnosis, although a larger fraction saying the government should outright ban this practice. And then moving into not yet possible technologies, but just for comparison, human germline genetic modification, you see the gray slice, the banded slice getting larger, and then not surprisingly, for reproductive cloning, everybody says, eh. So we can talk more about what the public's thinking about these technologies, the policy options, the report on what the American public is thinking about this, and also a description of a pretty unique experiment that we did to try to engage the public, not just ask them what they think, but really engage them in a conversation about reproductive genetic technologies, is back on the back table. And I'll turn it back over to you, Robert.
Thanks, Kathy. That it's always so helpful to start a conversation like this with some data, and the data you're showing seems to make a great deal of sense just from what one would observe in conversations, reading letters to the editor and editorial pages. Let me open it to the rest of the panel for any of your comments that have to do with either a sense, quantitative or qualitative, about public opinion in this domain, not just reproductive genetic testing, but other genetic testing as well.
It seems very likely that most people, and here I will defer to data if Kathy Hudson has additional data on this, but from what we've been able to learn at the Hastings Center, most people have a somewhat exaggerated conception of the connection between genetic information and most of the things that are going to be important to our lives. Now, there are some diseases where the connection is absolutely direct. In Huntington's disease, you don't get Huntington's without the genetic stutter that's at its root. And if you have the genetic stutter and it's long enough and you live long enough, so far as we know, you get the disease. So there really is a direct connection. Most of the genes that are being discovered for most of the illnesses, causes of premature death, and other kinds of social characteristics of interest, the relationship between the genotype and the phenotype is going to be very, very complicated. And my guess is most people assume it's going to be a lot simpler than it is. So there's this exaggerated view of the power and penetration of genetics into our lives.
Barbara?
Well, I totally agree with Tom that I think there is an inflated view of the power of genetics. But I'd just like to make the point that if you that this view, I think, is not limited to the lay public. I think that distinction is not particularly helpful, actually, in some ways, because I think the power of the paradigm of genetic thinking today is equally powerful across different scientific communities, and that's something that we have to account for, and it has lots of fallout in the way we do our science. For example, it leads to sometimes it may, I shouldn't say this in Washington, some NIH demon will come and get me, but if perhaps starting with the genomic approach is not the right answer for absolutely every single health problem that's facing the United States, or as we move toward issues that are more at the intersection of behavior and environment, et cetera, then that makes it even more problematic to be looking at genetic or genomic solutions, which isn't to say that they're not useful. And I guess the other observation I'd like to make is that even though if you press almost every geneticist, and every good geneticist will tell you when if you push them on this, that of course genes by themselves do almost nothing, except in the rare kinds of cases that we know about and that Tom Murray just mentioned. But if you actually examine the practice and the kinds of statements people make, there is a very naive sort of genetic determinism which keeps coming back into the discourse in spite of the fact that we all object to it. So, I mean, as an anthropologist who studies the way knowledge travels and moves across different domains, that's an interesting thing. Why is it that although we know it, it still has such social power? So I think that's an important thing to keep in mind.
Well, why?
I don't have an answer to that. I think it's because we want it to be true. I think we want it to be simple. The power, and I'll just make one hypothesis, and actually Stephen Jay Gould actually editorialized this when the genome was mapped, and basically said, oh, you know, since the Enlightenment, we have in science had a very faith in reductionist paradigms of inquiry. It's been the thing that's really helped us make progress. And now, you know, perhaps we're at a point where we need to move beyond that. So we're on the verge of moving to these very complicated quantitative ways of thinking. But those are very hard, and most people don't do it intuitively. So we I think, keep falling back into these ideas of determinism. But so anyway.
So a week ago,
Yeah.
I couldn't spell stochastic, and now I are one, huh?
Yeah.
Kathy?
Yeah, I think I'd like to separate it actually into three separate areas. One would be discovery, one would be preclinical, and one would be clinical. And I think there's been clearly a lot of exposure to the public, particularly in a lot of lay magazines with respect to promises of new technology. And so I think there's a lot of public expectation. So separating expectation from confidence. And I think that's an important point to make, because I think with any movement in science, there's the expectation, there's good news, there's disappointment. And I think from the very basic areas, which is the discovery, I think the new technologies, particularly gene expression, microarray, proteomics, are going to be key tools for new discoveries. And I think that is fundamental. I'm not sure how much, and I think the public, while not totally engaged in that, feels that that's becoming a part of the scientific discipline in order to raise a hypothesis and study a hypothesis from a scientific problem point of view. Then you get into the whole area of drug and diagnostic development. And that's, I think, has some very good examples where we've been able to actually, using some of these technologies, be they proteomics or gene expression microarrays, to identify populations that might be better candidates for a particular treatment, or we can detect a disease earlier and have better chances of maybe not curing it, but giving a longer lifespan with quality, and that particularly is true with cancer, and I think there's been a number of examples in that area. I think in the infectious disease areas, looking for targets for pathogens and developing new medicines for pathogens is clearly an area where there's a promise. If you take it the next step and looking at preclinical models where you're actually using some of these techniques, it's still at its early stage, and I think that will actually provide a lot of data which will be useful in developing better clinical trials for the utilization of many of these techniques, not only for diagnosis, but actually looking at toxicology and looking at pharmacology of some of these things. And finally, if you look way down the road and looking at opportunities for gene therapy, which is clearly, you know, we've had examples of hype in gene therapy and disappointment in gene therapy. But I think we need to step back and now go in a more systematic approach and be open with the public in both the positive and the negatives and try to do better science, both basic and clinical.
We had Anna Barker from the National Cancer Institute on an earlier panel, and she made the observation pertaining, I think, to your third category, some of the clinical applications, that after we had the success of Gleevec in addressing chronic myelogenous leukemia, there was the thought that, oh, this is the first of many, and they're going to start falling like bowling pins, but that hasn't really happened. Vince?
I would just like to raise some concerns about how much the public understands about genetic testing. I think Kathy's presentation talks about genetic testing. Well, really what you're looking for is a very finite set of information, a very finite set of data in the way it's performed today. That's going to change dramatically in the next couple of decades. The education of the public to bring them along from this very simple paradigm for looking at what genetic testing can offer them, they need to be brought along with that process. There's a great danger that they won't understand, the public, of which I am a member, will not understand the meaning of the information that's coming out, but even more dangerous is the fact that they won't know what they can trust and what they can't trust. Within the 15 minutes we've been sitting here, two genetic tests have been mentioned. The HER2 for Herceptin treatment for breast cancer, and the the triple repeat for Huntington's disease. Both of those tests are seductively attractive in being able to address a very important healthcare item. But both of them are not at the reliability level where you would say, oh, my cholesterol is this, I need to go get a statin. You know, we're far, far away in those two instances from being able to put the confidence levels on the information that we're getting in order to prescribe a particular therapy or a particular course of action in correcting health care. So those are the two major points that I think that the public really needs to come along with the science. That is, understanding the complexity of the information that's being fed, that's coming out of this process. And the other is, what confidence level do we have that we're getting what we buy?
Yeah, the fellow that broke me in as a forecaster, Roy Amara, had a wonderful observation. He said that with innovations, with new technologies particularly, we tend to overestimate their impact in the short term and to underestimate their impact in the long term. And you know, I think that's exactly what we've been talking about. You know, overestimation in the short term is maybe a long-winded way of saying hype, and hype and misunderstanding. And it sounds like we're at a stage there. I'd like to shift. Yeah, go ahead, Barbara.
Well, I was just going to say that there also are some structural issues that I think affect the fact that hype is so prominent, particularly in the U.S. context. And one of the things that I came away from the Secretary's Advisory Committee with was a real sense that one of the sort of unexamined problems is that sometimes the early commercialization of genetic tests are almost a byproduct of doing the science itself. And let me explain a little further. So back to our first session this morning where we were talking the question of RO1 versus ROI, or return on investment versus investigator-initiated award. We have a society where, and this is the nature of discovery in the U.S. context, where there's an enormous amount of pressure to take early findings and to try and commercialize them. And so that creates a real tension in that the hype and the promise comes partly from that commercialization process. It's then accentuated by the problem that there is no regulation of things like direct-to-consumer marketing of some of those early tests, et cetera. So I think it's not simply a matter of educating people to look out for some of these tests that get on the market. You know, I mean, it's also a real issue of making sure that we're clear about the context and why there are these tensions and why the hype is almost built into the process.
Kathy?
I want to respond to Vince's comment about how the public is brought along and whether they'll be able to sort of keep up. And I think that we've been, in the scientific community, deeply concerned about that for many, many years. And yet we haven't really explored, well, what do you do about it? We have various programs where scientists sort of talk at the public. and we haven't really evaluated how effective those are. So we haven't really explored mechanisms by which we can bring the public along. AAAS now has a new initiative in public engagement, and we'll see how that goes. But I don't think that we really have the tools yet to be able to know how to effectively engage the public and then to know whether or not that program worked.
Yes. I'd like to shift now and kind of work our way through the life cycle of a human being. And Tom, let me turn to you first to talk about some of the issues in newborn screening. I mean, presumably we are developing, we have and are developing tests for screening newborns for predispositions to diseases, conceivably diseases that we've got no chance of treating. Can you talk some about the dilemmas that we face there?
I should say that I think every significant point that's been made here, and they've all been significant, sort of come together in newborn screening and the issues that we've uncovered in the project that we're currently working on. If any of you have consumed a diet beverage that was sweetened with equal, which is a combination of 2 amino acids, one of them is phenylalanine, you'll have noticed this little print on the bottle or the can that says phenylketonuria, don't drink this. You know why that is? That actually is one of the measures of the success of the very first newborn screening program that began in the United States. It began for a disease called PKU, phenylketonuria. A child with that disease cannot metabolize fully the amino acid phenylalanine, and it ends up as a toxic metabolite, and this will cause neurological damage, retardation, behavioral disorders, and it's a bad disease. There's no necessity that someone born with this disorder end up with all the consequences of unprevented PKU. So we don't treat it medically. What we do is we just make sure that as a child you don't get phenylalanine, or you get it in such tiny quantities that it doesn't harm you. So children identified by newborn screening as being phenylketonurics are put on a special diet, and that turns out to be reasonably effective in preventing the consequences of the disease. This started about 30 years ago. Today, there are about 4 million children born each year in the United States, and virtually every single one of them is subject to newborn screening. In almost all states, Maryland, by the way, is an exception, the parents are not asked for their consent. The child is screened. This is a little heel prick that we take from every baby. We do a little capillary tube of blood, and we put a little spot on what's called a Guthrie card after its inventor. That blood spot is then sent to a lab. It's analyzed. Initially, it was for PKU. Now there are a number of other tests that can be done on it. And then information is fed back to the, usually to the physician taking care of this child, to the pediatrician. And if the child is identified in the screen, almost always there are subsequent tests to confirm what the screening test may have indicated. So we set the screening test to be very sensitive, right, so that we don't miss kids. That means it's not always very specific, so you get a lot of false positives depending on the particular test. All right, that's just a part of it, of how it goes. Now there are a few characteristics of newborn screening that are very important to bear in mind as we look at the challenges that it now faces. Number one, it's universal. As I said, very few children don't get screened. Number two, as a public health entity, public health program, we don't ask for your permission. We just do it. People generally don't object to this because they understand why it's done. It's just done to benefit the child. When, in a state like Maryland, when parents are asked, they overwhelmingly consent. People are happy to see this being done. Number three, the test of the program, if you think the test of the program is in gathering the sample and analyzing the sample, you are grossly mistaken. Those are the relatively cheap portions. It's what happens afterwards, because then after you've been identified in the screen, there's got to be confirmatory testing. If it's found out that you do indeed have this, well, one of two things are going to happen. Either the further tests are going to say you don't have the disease, so we will have incurred the costs of that, much more expensive testing, individualized testing, and will end up saying, well, there was nothing to worry about in the first place. Or you will have the disease, in which case there will be whatever is required in terms of ongoing treatment, special diets, drugs, whatever else, medical supervision, what's called the supervision of the medical home, which is often your home. All these, and these can be for decades, the cost. So the cost of gathering and testing are not insignificant, but they're a tiny proportion of the actual overall cost of an effective program of newborn screening. Now, what's happened today? What's happening today is a variety of things. Number one, we have new technologies, tandem mass spectrometry, for example. It's as easy to test for 30 disorders by tandem MS as it is to test for one. It's one run through the machine. The various DNA tests, like the gene arrays, one could imagine testing for hundreds, if not thousands, or tens of thousands of genes. Now, historically, the tests have been for inborn errors of metabolism, basically genetic issues, but the tests themselves have not been genetic tests. The tests have been for markers, metabolites often in the blood. We may be moving more towards, because of the advances in the technology, we may be moving towards a much more ambitious program of testing based on these little blood spots we get from the Guthrie cards. Is it a good idea? One reason the program has been such an unheralded success, newborn screening, has been that we've had very strict and pretty narrow criteria for what we would screen for. It would only be for diseases that wouldn't be otherwise discovered quickly, where one could in fact do something with the child to prevent or treat the disease and therefore spare the child the consequences of the untreated disease. Those are there were some others. They had to be cost effective. There were the Wilson-Younger criteria. There were about a dozen of them. Those are the key ones. So what happens now if we have the capacity with the same blood spot and the same machine at maybe no additional cost or minor additional cost? We could test for a variety of other genetic circumstances. I don't want to call them diseases because we don't know if they're diseases. In which some of them will be diseases, but we can't do a damn thing for your kid, at least not today. Others, we're not sure what to make of them. We know that, for example, for disease like MCAD, and I won't attempt right now the long name of that, that some people born with the mutation in this will be, can have sudden death, an episode of sudden death. Others will have a full lifetime completely unsymptomatic. What do we do with those? And what about cases where we get an interesting finding, it looks to be an anomaly, but we really don't know what its medical significance is? Two questions arise, at least two questions arise there. Number one, if we're doing these kinds of tests, not the straightforward PKU kinds of tests where we know we're doing something good for the kid, should we insist on informed consent from the parents, number one? And number two, is it even a good idea to present ambiguous information of this sort to parents? Information is not cost-free in the human sense. The additional medical supervision, et cetera, will be cost in a traditional economic sense, but also the worry, the concern, et cetera, that visited on the child will also be significant. I'm not offering an answer here. I'm saying these are the challenges that we face with this. There is a third challenge. Is this a wise use of public health resources at this point? So those are the sorts of challenges we'll wrestle with, and I will turn it to the next.
Let me push you beyond just posing the questions, though, to at least venture some opinion. I mean, it seems unmitigatedly good to test for the first 30 that you were talking about, and mainly, at least in this here, I'm talking opinion and not as an expert, mainly because of your second criterion, because you can do something about it. in those instances where it's ambiguous or where you can't, what's your take? And you know, I may be asking you to go beyond your professional take to a personal take on how we should make decisions about that, and whether, for instance, those should be included in a panel of tests for newborns.
You are pushing me, but that's your job as moderator, so it's okay. Our project is still in process, so I'm reluctant to say what the conclusions will be when we really haven't come to them yet. The challenge is very significant. I mean, there's no question that some of the strongest proponents, the most persistent voices in favor of providing maximum information to parents, even if it's clinically ambiguous, that's come from parents. Particularly parents who have had children who were not detected, who had some inborn error, genetic error, and who the parents at least hope, maybe correctly or maybe just hopefully, the consequences could have been prevented had they found out in the newborn time. The interesting thing, one of the interesting things about newborn screening is, of course, this totally gets us away from issues about genetic testing and abortion, right? Baby's born now. We're not talking about prenatal testing or even pre-implantation genetic testing. So we can put all that aside and just focus on what's good for the baby, what's good for the family, and is this a good use of social resources. It's also true that there have been there's been some pushback from public health experts who are very uncomfortable being pressed now to go from a role in which they felt very comfortable, you know, only giving information when we could do something concretely with it, to now being said just give information. And you know, there are intermediate positions. We could, for example, still keep the rule that we only, on the newborn screening that's done, still done without asking for parental consent. Let's say we keep that model. Only doing the conditions where we believe we can make a difference. And offering supplemental tests, either, you know, use the same collection. Parents could then decide whether or not to sign up for a supplemental test then, or they could sign up when they're, you know, early visits to their pediatrician. So there are other models intermediate. And I think right now there's a lot of wrestling and a lot of soul searching in this country about which will be the right one.
Good, thank you. Kathy, let me turn it to you to elaborate maybe on reproductive testing and some of the issues that arise there.
Tom talked about the tests that are available where there's something that you can do about it, where there is an effective intervention available. In the reproductive context, there's all sorts of tests that are available where there is no effective intervention and where the option is to avoid initiating a pregnancy, to avoid transferring an embryo to start a pregnancy, and making the decision to continue or terminate a pregnancy. And in that context, it's really important that the genetic test is undertaken in an informed way, in a non-coerced way. And it's a question, I think now, as new tests are becoming the standard of care in the reproductive context, how much people are really freely able to make those decisions. Then the question is, are those tests accurate? And how accurate should they be in order to be offered in the reproductive context? We don't really have a system. While most tests that are routinely offered in the reproductive context do have a high level of accuracy currently, we don't have any system of assurance that that's the case. So there is no review that takes place before a genetic test enters into commerce. And the determination of whether or not something is clinically available is whether or not the laboratory that performs the test is CLIA certified, which is an arcane, rather boring statute that basically says the laboratory's refrigerator is running and they have appropriately trained personnel. It doesn't say whether or not the test works. And then there's an interesting loophole where laboratories that perform tests on embryos are completely ungoverned because of a reticence on the part of agency officials to say that the statute, CLIA, that says laboratories that perform analysis of human beings for the diagnosis and assessment of human health are governed by CLIA. Well, they don't want to say that an embryo is a human being. So therefore, those laboratories, we don't even know if their refrigerators are running. So I think in the context of making fundamentally important decisions about the children that we have, that we might want to look at what opportunities there are for improving the confidence that we can have in those tests.
And I know in a little bit we'll come back to the mechanisms for the confidence on those tests, and there's a lot to say about that. But before that, Barbara, let me turn to you and ask you to comment on adult genetic testing.
If I may, before I turn to that, I just want to follow up one sentence each on Tom and Kathy in terms of sort of my take, and I think this will all come together. So one of the things that Tom didn't mention that is the sort of thing I think about in terms of the political and economic context in which we make these decisions is the fact that these new technologies, some of the impetus, I think, for the expansion of newborn screening comes not just from parental interest groups, although that's an important fact, but it also comes from companies wanting to be able to use these mass spec technologies broadly and to capture this particular market. And so
that's right, and the companies making the tandem mass spec machines have been lobbying to get wider use of them.
So a technology push rather than a demand pull.
Right. So we have to sort of think about why it is that certain social choices or decisions are happening, and and this is all done by the states, so it's pretty much a very complicated process. And then in the reproductive arena, one of the, I think, really difficult social choices that we face is the question of, even if we had an array of tests that we knew pretty well that they measured what they said they were going to measure, which is a huge complicated thing we'll get to, what panel of tests, what panel of genetic tests would we want as a society to offer to people who could use them in any of the contexts, pre-implantation, prenatal, and we really are not facing that at all. And I think I have some concerns about turning this all over to a sort of market-driven approach as opposed to making some really hard choices about which ones, and this may seem a little paternalistic, but actually having some sort of a process to come up to some social consensus, and that may be your next project. But at any rate, in the adult arena, that's really, I think, where the promise of this individualizing medicine through genomics comes in. And it was mentioned this morning. I mean, I think the ultimate idea of that is people think that everyone's going to be carrying around their little smart card or CD-ROM chip with their whole genome, and then everything that all their health care in the future is going to be governed by their own specific genes and what those genes will predict about their future health, about how they'll respond to diseases, about how they might respond to certain environmental insults. And it may be that we'll move there, but I think that there are some barriers to that we have to face. One is, I'll just start with the biggest one. The biggest one is the sort of collapse of the healthcare system in general, and the fact that, if you think about it, at the moment, we're not even doing a very good job of delivering non-individualized medicine. Even if we want to do the same thing for everyone, we can't seem to figure out how to do it and how to pay for it. So it might be good to sort of think about all those things at once and to be considering the healthcare system too. So another concern that I have in the area of individualized genetic testing is that it's based on a certain paradigm of thinking about how to do healthcare. And that paradigm is, I would describe as follows. The idea that you can predict risk and susceptibility to an event in the future in order to do something about it. Now, that's so commonsensical in the way we do healthcare today that it seems even almost wrong to question it. We test your cholesterol so that you can do something about it. We test various things. You might want to have if you're from a family with many people with breast or ovarian cancer, you might want to have a test for breast cancer risk susceptibility. And those things seem very commonsensical. Maybe we can expand this to cancer, to other kinds of cancer, to cardiovascular disease. But the one sort of unintended consequence that I worry about in that is that it seems to be moving us toward a future in which everyone's individual health becomes their own personal responsibility. And so health increasingly is going to become almost a market good in which how you deal with it, how you respond is you get your profile, you pay for that, and then out in the marketplace, you choose and buy the right set of products to keep yourself healthy. And that might fit into certain kinds of political regimes, but I'd like to point out that it keeps us from focusing on some other very important elements of health. In particular, a notion that perhaps some elements of health are really a social or a common good, which we have to look at in a different way. So those are sort of these are the fairly esoteric things, I'm afraid, that I worry about.
No, that's interesting. Your last point particularly, you and I both come from Northern California.
Uh-oh.
And in Northern California, I think that last condition that you described of, I am the master of my health, and
Right.
and between, me, my psychiatrist, my yoga teacher, and my urologist,
Not to mention the cosmetic surgeon.
not to mention, that's right, I didn't mention her, sorry.
Right. So I was just in Orange County where I opened up an ad. It was like a parody. There were 10 ads for various kinds of cosmetic surgeons, and I got a little concerned about that.
Yeah, that's right. Well, you've raised some interesting issues, and Kathy, I'd like to use the issue that you raised on testing of embryos not being CLIA covered as a segue to start to talk about standards for these tests and the assumption, I guess, that a member of the lay public would make that, gee, if I go out and buy the test on the market, it must be accurate. And Vince, maybe you can start the conversation about that.
Well, I'm not so concerned about the tests that are out on the market. I think, well, I am concerned. It's still a buyer beware issue with a lot of these things, and I think the powers that be recognize that that status quo is not acceptable, and that we're going to have to move beyond that. I was thinking about the best way to frame this, and I think what's lacking is we used it in earlier sessions this morning, leadership or political will, call it what you do. In the area of human identification and forensics application of genetic testing, that's all it is. There are 13 markers that will take the identification process out to about 5 or 6 nines. There's almost absolute certainty based on 13 markers. Now, it's been recognized for over 10 years that we can't make mistakes in a court of law. We can't afford to. And you find people incarcerated for most of their lives being set free based on genetic information. You find people being taken in for crimes based on genetic. It's a very well-run machine. And now, what happened there is that 10 years ago, the leadership, the political will was there to make sure that was tight technology, and the laboratories which do for fee the genetic testing for forensic purposes need to be certified once a year by the FBI, and the way they win certification is to show how their protocols, with the operators in place, the technicians in place who will run the test, that they can perform at a certain proficiency level. Round robins are run in a anonymous way once a year, and people who don't participate lose the license to do that kind of testing for whatever judicial district they're in. The breakdowns, when they occur, I don't know if anyone here remembers it, but about 2 years ago, the Houston Crime Lab blew about 100 cases. They had to turn 100 people loose because the DNA evidence was flawed. That laboratory did not go through the protocol, and it passed through the web of checks and balances, which should have called that that technician, most of it was assigned to a particular technician. But the point of that story is we can do it. We can get the same level of certainty, the same level of confidence into genetic testing for it. It's going to be, in some cases, more complex than 13 markers. And the way we sample, the way we handle the information, every step of the way is going to have to be validated and based on some performance metric. But again, I think it's going to take leadership and a will to do it, and there's no other better. We talked about the parent lobby for newborns. Where's the lobby for the rest of us? You know, for the implementation of this very seductive technology to make healthcare better.
Tom?
Somehow I'm not so reassured having heard your example about the Houston crime lab.
There will be mistakes.
It seems like we need not only the kind of political will to set the appropriate quality control system in place to begin with, which absolutely agree with you on that, but we need something like unceasing vigilance to make sure that, you know, the failures aren't terrible. A parent whose kid with PKU who doesn't get picked up because the lab tech had a bad day is a pretty frightening prospect. At least to me, as frightening as sending somebody to jail or acquitting somebody.
Kathy, anything to add to that?
Yeah, I think that there's, this is probably a nice segue into looking at not only the setting of standards and the adoption of standards that allow a certain level of confidence in the information. And I think it brings in then what is an appropriate level of regulation of these new technologies. And one is the tools that one needs to perform a technology. The second is the standards which one will perform the technology. And this can be looked at in terms of two things, both physical standards and written standards. And then you have the risk-benefit analysis for each of the applications of a technology and what is the impact that will have at the end. And there's no black and white. It's various shades of gray, so it's going to require probably a series of levels of different issues. And we've had experience, you know, at the agency, certainly when I was there, and I'm sure it's still going on, is when a test is developed, it's say a typical diagnostic is developed. If you're just getting a clinical test, there's CLIA that overlooks it. However, if you're screening the blood supply for a particular HIV antigen or an antibody, the level of scrutiny on that is much higher, the level of regulation is much higher, and the risk-benefit is such that what is the consequence of this if something doesn't happen? And I think it's an interesting point because most of what we are looking at now in the genetic testing and in proteomics is really multiplex testing. So the ability then to develop the appropriate standards as well as looking at what the information is going to be used for in terms of clinical decision making becomes very important, because if I'm going to make a clinical decision based on a test to do a gene therapy, and that's the only information I have, then I need a high level of assurance that that piece of information is very correct.
Let me ask you a question about standards. In one of the earlier panels, we were having a discussion of standards for electronic medical records and other information standards in healthcare. And the observation, I think it was Carol Kovac from IBM, made the observation that it's almost the inverse, I think, maybe of what you're talking about. Not that we don't have a lack of standards, but we've got a surplus of conflicting standards, all trying to aim at the same market niche, because a lot of them are standards that are driven by companies trying to get their technology accepted as the standard. Are there areas in genetic research or proteomic research where we've got this same kind of a phenomenon going on, where there are multiple competing standards, and that effectively this competition among standards is retarding the development of the field rather than promoting it as standards ought to be doing?
Well, I think you should take the first shot at this, Vince.
There. Takes a second for that green.
You can just leave it on. It's all right.
The easy one. Proteomics, there are no standards. There's nobody out there pretending they know how to do that. It's a very powerful tool used internally for drug discovery, where the process of data acquisition and management is entirely under the control of those who are seeking information about the value of a therapeutic or something. But it's, in my opinion, it's far, far away from needing a standard for clinical applications. The technology is just not ready. And you will get differing opinions about that. But now, with the area of genomic information, it's a little different. And one of the activities that Kathy and I have been involved in is the establishment of controls for microarrays, particularly those that are applied to gene expression measurements. And in that case, I like to think that her agency or the FDA, at least, and my agency, have stepped up to there are standards control materials which are being sold by reagent makers so that people doing research or applying these measurements can have some internal controls for their own purposes. But these are not necessarily the standards which will be required to regulate a clinical application or a microarray used for personalized medicine. That's going to take a higher order of oversight. That is, in my opinion, that's a natural role for government. I mean, it's health that we're supposed to be protecting. Now, we are on the verge of pulling it off, I think, through the External RNA Control Consortium, but it meant negotiating with several companies which are in this market of selling controls for research purposes. And of course, there are business plans out there that this process of normalizing the way we practice this technology are going to lose. They're going to have to give something up with the individual business plan that they have for that organization in order to co-opt for this greater good. But it's working, it's coming to pass, and I think the win for everybody is that their business plan will be revised, but in the end, all boats will rise. And so that's the paradigm that we're working under on that one.
Yes. Kathy?
I would just like to echo what Vince just said, because we're a little bit farther ahead in the gene expression microarray area, and I think the External RNA Control Consortium has really been a very good process, not only of looking at the physical standard, but also how to deal with the data that one gets from the information from the arrays, and I think that's very important. And it's also looking to see that that these standards can be used in real-time PCR as well, which I think is important to validate and complement the information from some of the array information. So I think that is going to be an important part of the process, and progress is being made, and I think it's taken a lot of effort on a lot of people's part, and I think it's good to see government agencies, academic groups, and private industry equally engaged in this. I would say that in the proteomics area, it is definitely at its embryonic stage, Kathy. You know, there's talk of standards. You know, HUPO, which is the Human Proteome Organization, has been speaking to this, but the complexity of the problem and the complexity, even just looking at databases, the correlation between information in databases is really a real challenge. And so I think it's something that the sooner that people can take a look at this in a very systematic fashion, I think the the better off we will be. But one of the things when we look at standards and establishing them and the validation, there's so many components. I think initially Vince talked about, you know, something so simple as sample preparation or shipping of samples can have major impacts on the validity of the results, either if you're looking at you know, gene expression microarray data or looking at proteomic profiles.
Let alone proteins that are even more volatile.
And so then you take, you know, you look at different kind of platforms that you're using, you're looking at different kind of software, bioinformatics, you're looking at different kind of probes, you're looking at different kind of labels. All of this are variables within a complex algorithm that goes into producing a result.
Good, thank you. I'd like to actually now come out to the audience and open it for your questions to the panel and comments to the panel. Anything that you would like to add?
Yes. Hi, thanks. I noticed in your list of Northern California individual health care providers, you left out medicinal marijuana prescriber. I wanted to ask, I was reading an article published a couple years ago by Baruch Brody, in which he calls for not only ethically but legally mandatory testing for certain genes which result in what he calls lives not worth living, and that is where it's terminal and the suffering is great. And he called for that to be put into law and for carrier testing actually to be linked to some good that society provides, like a driver's license. So that would be the time at which you would have to undergo such a test to determine whether you're a carrier. And I wondered, I noticed Kathy mentioned non-coerciveness in her view of genetic testing, but I wondered, you know, is there a growing opinion that there should be mandatory testing either at carrier status or within the IVF clinics even? As I guess there is, I guess the genetic tests of the newborns that Tom Murray was describing are mandatory. So I'd appreciate your reflections on that. And then just finally, Kathy, I wondered if in your survey and discussions with people, there was any sense that, oh, everyone should have access to PGD and IVF to deal with some of these genetic issues. Anyway, thank you.
So, to my knowledge, there has been no movement to force to have mandatory genetic testing, particularly in the reproductive context. There are medical guidelines that encourage and exhort physicians to offer certain genetic tests in the reproductive context. So women over 35, aneuploidy testing for Down syndrome, et cetera. And for all people of a reproductive age now, the American College of Obstetricians and Gynecologists recommends that people be offered or have made available to them, the distinction being a little fuzzy, CF testing, carrier testing. But the sort of the paradigm in genetics has always been that people have the right not to know genetic information as well as the right to know genetic information. And so the notion of people not having an option to say no really hasn't been seriously entertained.
Tom?
Yeah. I don't recall the article that you've described. It's hard for me to believe that Baruch Brody, whom I know well, would talk about a life unworthy of living in quite those terms. Lives not worth living. But I would have to second Kathy. There is a real distinction between the universal and therefore non-voluntary, which I guess is effectively equivalent to mandatory, except if a parent says, I don't want you to do this, they won't do it, right? So it is not mandatory, but the assumption is we're going to do this unless you object, except in Maryland where we get your permission. The difference is the point of the testing is to benefit that child who's being tested, right? So that's the public health justification for really pressing this program. Otherwise, any program of genetic testing, even for carrier testing, say, could be encouraged, but would have to remain, I think, fully voluntary. And I think most everybody in the ethics community certainly would say the same. Well, it's not universal, just like testing isn't universal.
My question, yes, Joy Almel here in American Cancer Society, so it's more of a cancer slant. But can we go back for just a minute to public confidence and maybe talk about physician confidence and knowledge? You may recall the MMWR that came out to evaluate physician knowledge and attitude as a follow-up to Myriad's DTC on BRCA1 and BRCA2, and the knowledge among physician populations was fairly low. And to compound this, we've been having some discussions with the American Academy of Family Physicians, and this is the first year ever in the history of AAFP that they've had the physicians push back on their annual clinical focus being on genetics because they don't want to learn about it. So could you comment how we'll address that gap?
Well, can I make one comment? It is absolutely true that physicians are having, and other healthcare providers are having, a really difficult time keeping up with what tests are available, how do you interpret those tests, how do you offer the right test to the right person at the right time. Fundamentally huge challenge for healthcare providers, especially given the sort of the compressed time in which they have to do what they do. The healthcare professional organizations are trying really hard to keep up. And so, like with ACOG developing the CF carrier testing standard with the American College of Medical Genetics, but there's a tiny handful of professional guidelines and practice standards and ethics statements for genetic tests, and a huge number of tests out there. And so somehow we need to bridge that gap between the number of tests that are being made available and being marketed and how prepared the healthcare providers are. And I think that the healthcare professional organizations are probably the right locus to really carry that forward, but they don't have the resources necessarily to be able to effectively do that. So that's a big challenge.
Barbara?
I just want to point out that to sort of to defend the physicians who are not wanting some of these educational sessions, something that I learned from Wiley Burke at the University of Washington is that it's generally because the way we do genetics education is especially for people like family physicians, is totally irrelevant to their everyday practice experience. And when you develop kinds of educational programs that actually tie the genetics knowledge into what they actually need in practice. So to give one example, so if you think about the fact that one of the main things that family doctors do is they have preventing colon cancer in their mind, and they have a lot of ways of doing that. Well, you could, if you start out by teaching them, a huge long lecture on the genetics of colon cancer, which is a tiny, tiny percentage of what they're doing, that's generally not very effective. But if you can interest them in the whole sort of array of thinking about things like, okay, you start with screening everyone, this is what you do with everyone, and then at this point, then you go down different algorithms, and that's when you move into genetics, that's much more effective. But we haven't worked out all the details of doing that, and quite frankly, I think there's been so many different organizations and pushes that education is the answer to all of this that, they're probably just getting a little fatigued by it. So, I have actually the same issue with the constant, public education maneuvers too, which are, I think have some of the same problems.
Tom?
Kathy Hudson informed me at lunch that when the genetic anti-discrimination law is passed, I will not be one of the people congratulated on its passage, because for some 10 or so years now, I've been heretical in saying that, look, we shouldn't be treating genetics, so all of the rest of our health information over here and genetics over here, as something that was somehow utterly different and especially dangerous and toxic. So what Barbara just said, I think, is intriguing. I think that's really, we should domesticate genetics. If we're going to be talking to family physicians about it, it should be, where does genetics fit within this larger question of how do I respond to the threat of colon cancer in my patients? How do I respond to genetics with respect to breast cancer for my patients? That, I think, is a much finer strategy.
Yeah, just to follow on from that comment, if you look at how you're going to interface all that information, all the genetic tests back to the primary care physician, perhaps a good example are the Framingham Heart Study risk equations, where what actually happens is there are various boxes that get scored, and to reflect the imprecision of the measurement, the quantitative value is not used, rather everything's categorized and then it's given a score and it's added together. So it gets put into a spreadsheet, it gets put into a pocket calculator, whatever, but it comes up with a number. And then in the end, that helps inform the physician what's appropriate. It also incorporates risk factors, environmental risk factors, and I guess eventually genetic risk factors, but it's imprecise, it's fuzzy, it's complex, but it gives a simple answer.
Melissa Alexander from the Exploratorium. I was intrigued by what you just said, Barbara, about public education and this whole thing. Can you please just say a little bit more about that? And
oh, yes, I always get in trouble if I say anything negative about public education.
No, please.
It's from the point of my perspective as a social scientist. If you see a social problem and your solution is that you need more education, it's a fundamentally conservative approach rather than to sort of get to the heart of the matter. So that's one concern I have about it. Also, on the genetic discrimination issue, when I was serving on the Secretary's Advisory Committee on Genetic Testing, we did a huge public survey to get a sense of people's concerns about in the field of genetics. And one of the observations that I made when we finally got this data back was, well, everyone was saying, what is their main concern? Well, it was about genetic discrimination. Well, it didn't take a rocket scientist to figure out that the reason the public was giving that information back is because that had been the focus of the educational program. So it becomes sort of a self-fulfilling property. You do this public education, then you do a survey to see what people think, and then they give you back the answer sometimes. But so it's, and what the public really will think is very complicated, and how that's going to develop is very complicated, and what people really need to know is hard to determine.
Well, panel, I am going to throw you a curveball and ask you to put your forecaster hats on and to think out 5 years. I'm looking at the cover of Science magazine today, which is entitled Genetic Variation, and is reporting on a very large-scale study of genetic variation across populations. And what I'm going to ask you to do is to predict the genetics-related cover of Science magazine in February of 2010. Third week of February of 2010. So what I'm asking you is think out 5 years and thinking about the march of technology as we see it, the application of those technologies into the domains that you're familiar with, and think about what will be newsworthy from a scientific point of view five years hence. Barbara?
You want to start at this end?
I will start at this end.
Well, in terms of this cover of Science, I actually, back to one thing I said in my introduction, which is that I think some of the social consequences of genomic information that can't be regulated or are difficult to regulate are going to be some of the most compelling and interesting. So, and the intersection of race and studies of human genetic variation, social ideas of race and genetic variation, I think is a very good example of that. And it's a problem, I think an issue that's not going to go away. It's something we've been working at Stanford very intensively, trying to get geneticists and social scientists and humanists who think about race in the same room to try and talk about these things. And that's wonderfully one of the things that happens in this issue of Science. So my guess is that in 5 years, those are issues, though, even though I think sometimes the scientists would like to say, okay, we've raised the issue of race and genetics, we've now solved it, we've talked about, let's move on. We've solved that. I think, unfortunately, it's one of the issues that I wouldn't be surprised if it's still on the cover of Science in 5 years, because I think I think that the social consequences that are about just how we see the world, how we understand the world, are the ones that are going to continue to be salient. But on the issue of genetics, I think that what the cover is likely to say in 5 years, someone might do a retrospective of saying how we actually did a bad job of predicting how quickly some of these new individualized medical techniques came in, and they'll be lamenting the fact that that we don't have as many breakthroughs as we had hoped, because my view is it's just going to take a lot longer to get some of these things into the marketplace.
Thanks. Kathy?
Well, I predict that the cover of Science actually won't be about genetics, but assuming it is about genetics, I think the place where we're likely to see the biggest shifts are not in the development of new drugs based on molecular understanding, but rather going back out to the drugs that are already out there and redefining who the appropriate population is who should be taking those drugs. And I think there's possibilities that will make big strides in that area in the next 10 years.
So pairing diagnostics with drugs for a more tailored therapeutic kind of...
minimizing adverse consequences, maximizing efficacy.
Yes, thank you. Kathy?
Probably similar to Kathy's, although I would probably take it just a little bit wider and probably look at another evolution in systems biology as a whole, of which genetics and proteomics are a piece, because it's actually the integration of those pieces. And so I'm looking at it from more the basic science aspect, not necessarily the deliver of individual clinical medicines, because I do think we're at the verge of a revolution, but it's not quite here yet. We're trying to feel our way about this area of systems biology and its perspective and integration in a three-dimensional level. So I think that's my prediction.
Thanks. Vince?
Well, I'm glad Kathy brought up systems biology, because I think, but I don't know if that's ever going to be cover material. It's going to be the slow, persistent integration of multiple sources of information. Genomics, after all, we got there by being reductionist. But when we try to deliver health care better than we have in the past, it requires that we get out of that mode of thinking and start to integrate information back up the biological hierarchy. And I think that really is what systems biology is all about in many ways. But if, science and nature both are standard reading for myself, and I think 5 years from now you might see, instead of a pull-out of a new genome, which we get about every month out of one of these two magazines or not, it's very likely that over the next 5 to 10 years we're going to see pullouts of sequences which identify a particular condition of human health. It's not going to be a single SNP or a short. It's going to be multiple patterns of genetic information, which proteomic information will come along with it, along with gene expression. And I think we're going to see an exciting evolution of new conditions being identified with these different, and I'm just going to use a term, biomarkers. They're all biomarkers, and I think it's going to be a very exciting decade, but I'm not sure what covers are going to be.
So putting together the collections of biological materials, which we're calling biobanks, analytic platforms such as gene chips, which will now have more standardized ways of understanding and recording. New ways, powerful statistical techniques for studying and unraveling correlations between biological markers, biomarkers, genetic, also environmental correlates of complex disorders. Putting all those together, I'd love to see in 5 years an article that, on the cover of Science, that explains in a very powerful way the various genetic, environmental, and developmental contributions to a disease like diabetes, some other autoimmune disease, heart disease, or some such thing. That would be my hope, and that seems to me not entirely a fantasy.
Wonderful. Well, panel, I'd like to thank you very much for spending an entertaining and elucidating afternoon with me here. Tom?
Yeah, I think there's one more thing that needs to be said.
Yes.
Robert Mittman this is his 4th panel of the day.
Yeah.
This is a marathon, so can we also include him?
Thank you. Thanks. I've had more fun today than I can tell you. And so panel, thank you very much for the job well done.
Thank you.